In this video, we're going to start talking about neural signaling, meaning how neurons communicate with each other. And very broadly, we say that neurons can send electrochemical signals. And the electrical and the chemical part of that are two different things. The chemical part of it is happening at synapses, right? We haven't really got there yet, but these synapses are just connections between the axon of one neuron and the dendrites, or the cell body, of another neuron. So at those synapses, that's where we're going to release neurotransmitters. The neurotransmitters are the chemical part of electrochemical signaling. The electro part of it, that happens down the axon of a neuron. So if you get enough neurotransmitters that are excitatory, right? Neurotransmitters can either be excitatory and stimulate the cell, or they can be inhibitory and prevent the cell from firing. So if you get the release of enough excitatory neurotransmitters at all of these synapses across this neuron, you can get something called an action potential that travels all the way down the axon of that neuron. And an action potential is really an electrical signal. So we should probably have a quick discussion of what an electrical signal really means. When someone plugs a power cord into a wall, and we say that electricity flows through a cable to charge your phone battery or power your computer, it's electrons that are moving down that wire. And electrons are subatomic particles that have a negative charge. But really, electricity is any movement of anything with a charge anywhere. And so whether it's an electron moving, or it's sodium ions or potassium ions or chloride or calcium, it doesn't really matter. So these minerals are what cells use to send electricity, because they have charges and we can let them move back and forth across membranes. So when we say that an action potential is an electrical signal, it really literally is electricity. Ions moving across the membrane of this axon are going to send an electrical signal that's not really different in concept than electrons moving down a wire to send information between computers or any devices that are linked together by a conduction system, right? And so big picture, if we release enough excitatory neurotransmitters on the chemical end of things at the synapse, we get an electrical signal down the axon. Now, the catch is this action potential is a yes or no thing, right? We say action potentials are all or none. They either happen or they don't. So it's kind of like flipping a light switch. You either flip the switch on or you didn't, but there's no such thing as halfway flipping on a light switch. The lights don't come on half their brightness. It just doesn't work that way. Now, a dimmer switch that you can slide up and down, that would be different, but action potentials just don't work that way, right? So in order to set this up and have a discussion, we need to talk about something called membrane potential. And membrane potential, the term potential, think potential energy. The way this works is in all cells throughout the body, but especially neurons, we get an unequal distribution of ions across the cell. So what ends up happening, and we'll explain why in a bit, is you get chloride and sodium ions primarily built up on the outside of the cell, and you get potassium ions and a bunch of proteins and other things that are negatively charged built up on the inside of the cell. And what you can do is use something called a voltmeter, that's this guy, to measure the charge difference across that cell. So just like an electrician might use a voltmeter and stick the end of it in an outlet in your house and measure how much electrical current can flow through the socket, you can do the same thing microscopically with a very tiny little probe that you insert the tip of right into the inside of the cell. And what we're doing is we're measuring the difference between positive and negative charges on both sides of the cell. And the way that it works, for a couple of reasons we'll get into, the inside of the cell tends to be net negative. If you put this electrode in there, the inside of the cell tends to measure around negative 70 millivolts, right? You measure electricity in voltage. Milli just means thousandths of a volt. So we're talking very, very tiny amounts of electricity, but it is real electricity. All right. If the outside is negative 70, you can say that, the inside of the cell is negative 70. You can just as well say that the outside of the cell must be positive 70. It's just we measure the inside. And so when you see it on a graph, you're going to see that negative value from the inside of the cell. All right, now, what these wedges and triangles are showing you is the direction of these gradients, right? Chloride is pointing from high to low in this direction. Same thing with sodium. Remember, ions, if you let them flow, they tend to go down their gradient. Potassium is pointing high to low, the opposite direction. Right? And it's not just concentration, charge matters too. So we say this is an electrochemical gradient because not only does sodium want to go this way because it's from high to low concentration, the positive charge in the sodium is also attracted to the negative charge on the inside of the cell. Right? For potassium, its gradient is sort of conflicting. The high to low, the chemical part of it is pointing this way, but potassium really likes being attracted to the negative ions on the inside of the cell. And so in the end, in this case, the concentration gradient wins. And if we let potassium move through the membrane, it will flow this direction, and sodium and chloride will flow this direction. Right? So you have to consider concentration and charge, but in most neurons, most of the time, concentration wins. They flow downhill. All right, so why does this matter? In order to send electrical signals, we have to have that concentration gradient. And this sodium-potassium ATPase is an active transport pump that's a big part of making the gradient happen. So if you remember from a previous class, active transport means that you have a membrane transporter, right? It's a protein, that uses ATP to pump molecules places that they wouldn't normally want to go, molecules or ions. If this was a facilitated diffusion channel, there would be no ATP and things would just move from high to low and they'd hit equilibrium. That doesn't happen here. Our pump uses ATP to make sure that sodium goes outside of the cell, and it happens to pump 3 of those sodiums at a time. And same pump pushes potassium 2 at a time into the cell. So our net movement, we get 3 sodiums that leave the cell, we get 2 potassiums that enter the cell. That is a huge part of what's giving us our difference, right? More potassium in here, right? More sodium out here, right? If you didn't have this pump, the neuron would die immediately, or it wouldn't be able to send electrical signals anyway, because without that gradient, later on, we're going to start opening channels. And if you didn't have a gradient, nothing would happen, right? Ions won't flow through their channels if they're at equilibrium. And so we have to build sodium and potassium up unevenly across the cell. It's the same concept as why we pump water up a water tower. That way, if the power goes out, we've got a bunch of water that's higher than everyone's houses, so that when they open their faucet, water flows downhill. This is the same deal. We want sodium up here so that eventually when we open a channel that will let sodium flow through, we know that it's going to go across the membrane. And remember that anything with a charge, and these sodiums are positively charged, if it moves across the membrane, that's electricity. Because remember, electricity is just the movement of anything that has a charge. Okay, so when we're calculating this membrane potential in voltage, what matters? This equation is not nearly as bad as it looks. So all of these values that we're using to try to calculate our resting membrane potential, most of them are constants. R is the gas constant. Z is the ion valence state. So for sodium and potassium, it'd be +1. For chloride, it'd be -1. So that's pretty much set and determined. F is Faraday's constant, which is set and can't be changed. And temperature in a human doesn't really change because we maintain our body temperature. So that's kind of out of the equation too. Ln is just natural log. That's a function on your calculator. And so all that really winds up mattering is the concentration of ions outside the cell and the concentration of ions inside the cell, right? And so this Nernst equation looks really intimidating, but all it's telling you is we're counting the ions that are either on the inside or the outside of the cell and comparing them. And if we have more negatives on the inside, we say that we've got a negative resting membrane potential. And if there were more positives, then it would be positive. And so when we draw graphs eventually that are going to have membrane potential on their y-axis, that's all we're doing. We're asking which side of the cell is more positive and which one's more negative. Right. The other thing you may have thought about is if this pump is sending sodium out here and potassium in here, well, they both have positive charges, so does it really make a difference? Like, shouldn't they just cancel out and the membrane potential would be 0? The only reason that's not the case is because of these things that we call potassium leak channels, right? And leak channels just tells you that you have these potassium channels inserted in the cell membrane, and they don't really have to be open or closed. They constantly leak low amounts of potassium, right? These blue circles in our illustration down here can represent potassium ions. And so if the cell is constantly leaking a little bit of positive charge this way, that's what's going to make the membrane slightly positive on this side and slightly negative on that side, right? And I know that this doesn't seem like that big of a deal, but if these leak channels disappeared, a person would be brain dead instantly, right? You have to have that charge difference or you can't send electrical signals. Okay, so now that we've got our membrane potential set up, we can start asking, okay, how does a neuron change that membrane potential to actually send signals, okay? And that's going to be the topic of our next video. So follow the next one in the playlist. We'll start, branching off from that resting membrane potential, talking about graded potentials, action potentials, and how we actually get that cell activated to send the next signal down the line.